Now I'd like to introduce today's speaker, Dr. Leor Weinberg. Dr. Weinberg is director of the Gladstone Center for Cell Circuitry at the University of California, San Francisco, and is a distinguished professor with appointments in the Department of Biochemistry and Biophysics, as well as the Department of Pharmaceutical Chemistry. Dr. Weinberg is a highly successful and recognized virologist. He has made major contributions to efforts to reactivate latent HIV, which makes the virus more sensitive to antiviral drugs. Of particular relevance to today's webinar are his ideas and work on so-called therapeutic interfering particles. Within the context of this webinar series, one might think of these as essentially genetic biocontrol of viruses. Therapeutic interfering particles with an R0 greater than 1 will drive or spread through populations. Such particles have unique potential to help address particularly refractory problems in the HIV therapeutic space. We're going to hear more about such therapeutic interfering particles and the recent progress in their development and testing. Thanks, Leor, for participating in the webinar series. Really appreciate you taking the time, and we're looking forward to hearing about your work. At this point, I'm going to turn the floor over to you.
Great. Well, thank you, David, for this invitation and opportunity to present our work. Let me just move this over. Okay. So we work on antivirals. And one of the concepts that we've been working on for many years now, many decades now, is this antiviral technology we call therapeutic interfering particles that David introduced. These are essentially molecular parasites. This cartoon here demonstrates briefly how they work. I'll go into much more detail, but they're essentially engineered molecular parasites, fragments of viral RNA that compete with steal componentry from viruses. In this way, they conditionally replicate and generate viral-like particles, which are non-pathogenic. That's encompassed here in this schematic. And because they have the ability to replicate, they fall under this class of genetic biocontrol and have analogies and really similarities to gene-drive technologies. In fact, as I was telling David, the original paper on TIPs, or therapeutic interfering particles, was published in 2003, just a few months before the paper that popularized the gene drive concept, for me at least, Austin Burt's paper. So this is an old theory that we've been working on for quite a while. It was controversial at first. In fact, one of my colleagues referred to TIPs as a holy grail of virology, and I think he meant that cynically, as in you're never going to find these, but I'll show you that they exist. They are therapeutic, and they're now entering clinical trials. We have a clinical trial for both HIV and SARS CoV-2, and I'll tell you a little bit about that. So while we've been working on this concept for about 2 decades, it goes back much farther. Well, okay. It goes back much farther. Why is that not working? There we go. Really back to this individual, Preben von Magnus, who was a Danish microbiologist. He'd actually discovered monkeypox. And when von Magnus and colleagues were working on influenza viruses, they saw this effect that von Magnus lent his name to. When they infected tissue culture with isolates, they would first see the virus grow to very high levels and then essentially crash down to very low levels and recover. And this oscillation would continue, sustained. Von Magnus made the analogy to predator-prey dynamics, Locke-Volterra equations, and said what he thought was happening, hypothesized, was that there was very early on emergence of an incomplete virus, essentially a predator or parasite virus, which competed with the wild type and required the wild type. So it would compete with the wild type, feed off of it, and knock it down But when the wild type got to very low levels because of that competition, there was nothing left for this incomplete parasite virus to feed off of, and the wild type would recover, and this would continue, so on and so forth, these boom-bust cycles. So von Magnus called these incomplete. Others called them von Magnus particles, probably in jest, because he had no direct evidence for these particles, some indirect biochemical evidence maybe. And it really took almost 20 years for the first direct evidence from Adeline Hackett, who was a microscopist actually at the NIH and Berkeley. And she was studying VSV in culture. And typically VSV forms these uniform bullet-shaped particles. And this is the important part of uniform length shown here. But when when Hackett looked at these von Magnus oscillations, these cultures that were oscillating, she saw these short, stubby particles shown here and here. And this was really the first evidence for these incomplete particles. They had a mutation in the capsid protein, and they were short and stubby. Unfortunately, everyone forgot about Hackett, and what they remember is this very influential paper from David Baltimore and Alice Huang, where they renamed these into defective interfering particles, or DIPs, and implicated them in disease processes. And this led to a real influx of interest into the field in the 1970s. Many people tried to target defective interfering particles, or DIPs, with therapeutics, or use them as therapeutics. Unfortunately, the technology didn't exist. This was irrational exuberance. And the field really fell flat after the 1970s. And for a good 20 years, there was very little activity in the field. So this is where we come in, a series of actually mathematical models that we published in the early 2000s, 2003, when I was a graduate student, got the attention of some funding agencies, and they injected a huge amount of capital into the field in the early 2010s, and funded about 20 labs around the world, including us. And so I'll tell you about that. So here's the outline for the talk. First, I will tell you about the theory of a TIP and why we renamed it, or renamed it from a DIP to a TIP. Really, TIPs are a subset of defective interfering particles with an R naught greater than 1, engineered to have an R greater than 1. Then I'll tell you about the technology that we developed to screen and discover these TIPs for HIV and Zika. And then the first published TIP was for the SARS CoV-2 TIP just about 2 years ago. I'll tell you about the mechanism of action, the data in hamsters, and transmission. And so I'm going to focus largely on the underlying science for this technology, but I'm happy to answer questions about the implications, the ethics, the regulatory aspects. And I want to warn you that I tend to give long introductions. So 15 minutes from now, if you still haven't seen a piece of data, don't be alarmed, it will come. I always begin with my acknowledgements. Over the past 20 years, a lot of people in the lab have contributed to this. The hero, though, is Sonali Chadaverdi, a postdoc in the lab. She's the first author on the SARS CoV papers and co-corresponding, and she's on the job market, doing quite well. But if you are looking for a really sensational, off-the-charts person, she's available. I also got to thank Davey Smith, who's our clinical trialist at UCSD. He runs the clinical trials for HIV and for SARS CoV-2. And then Rob Roddick is a longtime colleague of mine, and we started a small biotech called VX Bio because Gladstone, my institute, cannot act as an IND sponsor to the FDA, so we had to start this company. Actually, at the behest of the Department of Defense, VX Bio was started in 2020, and of course the funders. Okay, so why did we go about building or forwarding this concept of TIPs, of these molecular parasites? And there was really two reasons, two unmet medical needs that we focused on. This is a very well-known piece of data from the CDC, actually, on annual influenza vaccine efficacy over the past almost 20 years. And you can see some years we do well, and the vaccine efficacy is 50% or so, or greater. And other years, because of genetic drift, the vaccine is not nearly as good. The guess is not nearly as good, and the vaccine has very little efficacy. For instance, 2008, 2009, 2014, and last year, the efficacy was also in the teens. And this is because the virus mutates. We call that genetic drift, and in some cases, shift. The vaccines, we have to guess every year how the virus is going to mutate. We're seeing the same issue now with SARS CoV-2. We have to update the vaccines because the virus is rapidly mutating, about 3 times faster than influenza. And for some viruses, the genetic diversity, the genetic variation is so great that we don't even have pictures like this. We can't even build a partially effective vaccine. HIV is one of those viruses. Herpes virus is another. So for HIV, we don't have a vaccine because the genetic diversity is just so enormous. For those viruses, we have very effective antivirals, but we have other problems that I'm going to group under the umbrella of deployment challenges. So we, for the past 2 decades or so, have initiated large-scale, continent-wide antiretroviral drug campaigns in Africa. These drugs are incredibly effective, very safe, and yet this is the current picture in sub-Saharan Africa. The last year that we have reliable data for, pre-pandemic, 2019, there were 1.6 million new infections. That's despite the antiretroviral drugs being widely available. In some parts of sub-Saharan, of South Africa, north of Durban, for example, near the Mozambique border, where I visited a few years ago, the lifetime likelihood of a young woman of childbearing age acquiring HIV in her lifetime is upwards of 90%. And that's despite the availability of drugs. So we know we have to do better. We know that drugs are not going to end this pandemic. And the question we had was, can we do anything about these 2 unmet needs by reaching back to the old idea of defective interfering particles? Since they can replicate, can they be vaccines or antivirals that have the ability to be single administration and overcome genetic diversity? And that was the idea. So we had this idea in the context of HIV. That's where I'll show you the idea first. So this is the genome of HIV. It's about 10,000 bases, or 9,700 base pairs from end to end, from the flanked by 2 LTRs, or long terminal repeats. The long terminal repeats, the 5' long terminal repeat encodes the promoter. There's a single promoter, which drives 9 genes. So GAG, POL, ON, they're shown in the brown color here. The promoter is very weak. Lentiviral promoters are very weak. But one gene, TAT, which we've worked on extensively, feeds back and transactivates the promoter, forms a strong positive feedback loop. And we characterize this very extensively. And when TAT is present, the virus promoter is very strong, and the virus transcribes. The RNA, this is the post, I'm showing you on the bottom, the post-integration life cycle of the virus. After it integrates, so the virus reverse transcribes its RNA into DNA, and that DNA integrates into the genome, the host genome, shown here. And when TAT is present at high levels, the virus can have high-level transcription. That RNA is exported from the nucleus to the cytoplasm. And those RNAs translate the proteins. So the purple dots here are the proteins. And if those RNAs are carrying this Greek symbol, this packaging signal we call STI, which is an RNA loop structure, stem loop structure, that RNA can be packaged in its own capsid and envelope and produce viral particles. So this is the life cycle of HIV in half a slide. Most viruses have a very similar life cycle, except for the DNA component. The retroviruses, lentiviruses are unique in that fashion. And so what was the idea for a defective interfering particle? I should point out that lentiviruses are the exception in the virus kingdom. They were the only family of viruses where we didn't have examples of defective interfering particles. All other viruses seem to. But we thought what it would look like would be very much like the old von Magnus Hackett defective particle, where you have a mutation in the capsid protein, or GAG. For example, this virus would not be able to replicate on its own because it doesn't have capsid. But if it's in the same cell, it can be complemented in trans by the wild type. It can steal what it needs from the wild type virus in the same cell. And we hypothesized that we could take this to the extreme and cut out all of the genes of the virus. And we're left with a small piece of genetic material, RNA, that we call the therapeutic interfering particle. And I'll show you why in the next slide. This could not do anything on its own because it doesn't have any of the genes coding for any of the viral componentry. But if it's in the same cell as the wild type virus, it can steal what it needs, parasitize the wild type. For instance, it can take TAT and transactivate its promoter, Rev, and export from the nucleus, for example. So if we could introduce this, the hypothesis was that we can introduce this into a cell by gene therapy approaches, which are very standard at this point, in blue here, it would start in the same cell replicating its RNA or transcribing its RNAs. And if we've engineered it properly, we could produce more of those RNAs. And because each of them has a packaging signal, there's a pool of a population of those RNAs that can stoichiometrically outcompete the virus for its own capsid and envelope. And what you have coming out of the cell are viral-like particles in blue here, which phenotypically have all of the proteins of HIV because they don't carry their own. So the capsid is from HIV, the envelope is from HIV, as you can see here. But genotypically, they're carrying the therapeutic interfering RNA, TIP. So the reason this was therapeutic is because it reduces viral load or viral concentration in the blood. And we know that viral concentration in the blood is the single best determinant, still remains to this day, the single best determinant of time to progression to disease and transmission potential. The higher the viral load, the more likely someone is to transmit. The lower the viral load, the less likely. And the higher the viral load, the more rapidly they progress to disease. The very avant-garde idea was that because these tips have their own viral load, they may be able to themselves transmit as well. And so the therapeutic would itself potentially transmit. I won't really talk about that so much, but there is a talk online about that concept if you'd like to look at it. So why did we rename this a therapeutic interfering particle? So let me go into one slide on the theoretical requisites of a TIP versus a DIP. So here, the wild type virus, in this case HIV, is shown in red here, and the DIP or the TIP is going to be shown in blue, light blue and dark blue, or cyan and purple. Okay, so the classic von Magnus DIPs have to satisfy two criteria. The first criterion is conditional replication. So the defective interfering particle can infect the cell, get in, but it can't do anything on its own. It will be dead end. The only way for it to replicate is if the wild type virus superinfects that cell, and then the DIP can be complemented in trans and replicate. So that's criteria number one, conditional replication. The second criteria is interference. The DIP has to actually do something, so interfere. So the wild type virus infects a cell and produces a burst size, we call it, a number of viral progeny. If the wild type virus infects or superinfects a cell where the DIP is already present, the burst size is lowered, reduced. So that's the interference criterion. And we introduced a third criterion, a quantitative criteria, that the R0 had to be greater than 1. The basic reproductive ratio had to be greater than 1. I used to have to explain what R0 was to people. Now we're all armchair epidemiologists after COVID, and everyone knows what R0 is. It can be defined, R0 can be defined at multiple scales, the population scale or the single cell scale. And here we used it at the engineering scale, the single cell scale. And our definition was that the number of a progeny from a single infected cell, the number of infected cells produced from a single DIP transduced cell had to be greater than 1. If that was true, if the DIP could produce more than one daughter transduced cell, the R0 would be greater than 1, and it would be stable. This is essentially a stability criteria, a bifurcation parameter. So that was the definition of a TIP, that it had to have this added criterion. all TIPs are DIPs. It's just that not all DIPs are TIPs. So this was a very straightforward hypothesis to push forward, to test mathematically in computational models in the computer. Exceptionally difficult to engineer, and we failed for 20 years and over and over again. The only thing we were really able to do during that intervening time is test if we could build this R naught greater than 1 TIP, what would be the effect? Is it actually worth even trying to build it? So I'll show you one slide on what kept us going for 20 years, almost 20 years. And this is a well-established data set from Malawi that many groups use to project the impact of interventions. It's a UNAIDS data set, an antenatal clinic data set, where women went into clinics in the early 2000s who were women who were just given birth and were tested for HIV. And it was used to longitudinally follow prevalence in Malawi. At the time, in the late 2010s, in the late 2000s, early 2010s, the prevalence in that part of Malawi was almost 30%, very high. And it was believed to be stable, so that it was not going to get worse over time. And that's proven relatively true, I think. Many groups use this data set to predict the efficacy of antiretroviral therapies, or so-called universal test and treat approaches. We know these antiretroviral therapies, or ART cocktails, are exceptionally effective. And if we could get them to, let's say, 3/4 of the population, highly optimistic, this would be the reduction in prevalence from 30 down to somewhere around 20%. So somewhat sobering. We looked at the effect of vaccines. We still don't have an HIV vaccine. There was thought to be a 30% protective vaccine back in the early 2010s called RB144. Whether it's 30% protective, it's not clear, but we proposed if we could get this to 80% of the population, what would be the effect? Also quite sobering. A 50% protective vaccine, which will be deployed tomorrow if we had, the effects are, again, sobering. So we introduced TIPS into this model. So let me say that this is best case scenario. We know the virus mutates rapidly, overcomes monotherapies in a week, dual therapies in a few months, and in some cases can overcome triple drug therapy as well. So we introduced TIPS into this model, the TIPS that we thought we could produce in the laboratory, that they could reduce HIV viral load by 0.5 log or 1.5 logs, and the effects were very dramatic. I'm not going to tell you very much about this mathematical model, but this is what kept us going. We thought if we could actually build this, it might be impactful. So I'll give you one slide about why it's robust to evolution of the virus, why we think. Viral genomes, retroviruses in particular, produce a swarm of genotypes. Every replication event leads to an error. The error rate is about 10 to the fourth. The genome size is 10 to the fourth. And so in every individual, every day, you have a quasi-species or swarm of viral variants being formed. The dominant is the wild type. That's the one that replicates the fastest and is the most abundant. And the drugs essentially knock down that wild type quasi-species variant. But we know that resistant variants can arise that can emerge that are resistant to those drugs, and they're selected for. So that's the evolution or selection of resistance for many drugs, for many viruses. TIPS, because they're replicating using the same machinery, the reverse transcriptase of the wild type virus, they will have the same error rate and produce their own quasi-species or swarm when they knock down the virus. And if there is a resistant variant that even can arise, and we're not sure it can happen, the models show that there will be a reciprocal variant in the quasi-species of TIPS that should also be able to compete with and parasitize that resistant mutant. So this is the robustness. If you can introduce replication into the therapeutic, into the molecular parasite, you can set up what's called a co-evolutionary arms race. And that's what we think is occurring. I'll show you the data for that. Okay, so let me give you just a few slides on the technology we developed here. So this is a very long story, and I don't have time to go into it. It's actually a beautiful story, I think, of failure. We tried two approaches. The first approaches for the first 10 years or so were what I call the directed, educated guess approaches on the left here. HIV is arguably the best studied 9.7 kb ever. We should be able to just cut out the elements that we need and produce a TIP or a DIP very easily. And we tried that over and over again, and we failed. We were always able to cut out the components and produce something that interfered. We were never able to produce something that had an R naught greater than 1. So that was the failure mode. It was could never mobilize with R naught greater than 1. And so in about 5 years ago, 6 years ago, a very talented bioengineering student, Tim Nodden, came to the lab and convinced him to set up a high throughput screen where what Tim did is take the HIV genome, introduce a retrotransposon, which randomly integrates into the plasmid, which is a molecular clone of HIV. This randomly integrating transposon had meganuclease sites that could be excised from the plasmid. And so what you produce then at step 2 is a randomly positioned cut in the DNA shown here in the molecular clone of HIV. Then you can open that cut up with an enzyme blend in a regulated fashion, so you can make large cuts or a large open, increasing the size of the cut or smaller ones. And then you can recircularize the barcode, and what you get is a library of randomly positioned cuts of variable size. So deletions. And this is what the deletion library looked like. He produced a library of about 150,000 variants. I'm showing you a subset of that library here. The deletion coverage by deep sequencing on the y-axis and the HIV genome across here on the x ordinate. And so you can see we were able to make deletions pretty much everywhere in the genome. Right here at 12,000, this is the origin of replication of the backbone and the antibiotic resistance marker. There were no cuts or no deletions or barcodes in that region. That's essentially proof of sanity, because if there are any deletions in that region, the plasmid won't grow in the bacteria. So we won't be able to even make the deletion variant. There was a preponderance of barcodes or deletions in the 5' end of envelope, as you can see right here. And it turns out that envelope is highly cytotoxic, even in bacteria. So the bacteria that cut out the 5' end of envelope survived were more efficient and could replicate more efficiently. And that's why it's overrepresented. Otherwise, it's across the genome. So Tim took this library and passaged it in culture in the presence of HIV, and asked after 12 passages what was left, and he found that there are regions where there were no deletions after 12 passages. So the LTR, you need the 5' LTR promoter, the RRE or the Rev responsive element. These are 2 known sites, and then this site, the central polypurine tract, this was controversial in the field. It wasn't clear that it was required. I mean, it should be required because the virus maintains it, but its function was very controversial. And Tim found that actually the CPBT is as important as the promoter, the LTR, or the packaging signal or other sites. So we thought we had it. We thought this was the missing link. We just have to put the CPBT back in and we'll have a TIP. So Tim put that CPBT back in. We built 50 or 60 different molecular clones, and every one of them now replicated passage after passage, and not one of them interfered anymore. So it was a debilitating moment, depressing moment. We thought we could either build a therapeutic that could interfere or that could replicate conditionally, but not together. I actually thought we'd proved that this was an impossible notion. But at the same time, in the back of the lab, we were running this reactor. This was back in 2018. And this is essentially a chemostat for viruses, or a bioreactor. And the reason we built this was for a completely different reason. It was once we had a tip that was a foothold, the idea was to evolve it to be better and better against the virus. And to do that, we thought we needed bioreactors. And so we were running this bioreactor, but we didn't really have a tip yet. And so it wasn't clear to me that this was actually a useful exercise. But some of the students in the lab and postdocs went in and analyzed what was coming off of this bioreactor. And what they found was that this is a GFP expressing HIV. They saw these oscillations in the GFP expression. And they thought, well, maybe these are these von Magnus oscillations, perhaps. And they went in and analyzed the viral genome that was in these oscillations, and they found this viral variant called F1, which had a 2.5 kb deletion, and that deletion was right in the Pol VPR region. And it turned out that this was the first tip. We had to engineer it more because the CPPT was cut out, so we had to reintroduce the CPPT here. But once we did that, we had our first tip. And we synthesized this tip, and I'll show you a schematic of how it acts. So here we put a blue fluorescent protein into this tip, this deleted variant. We have an HIV with a red fluorescent protein, and the experiment, I'm going to show you the 40-hour movie where the cells, these CD4 T cells, were first infected or transduced with the therapeutic interfering particle, or the TIP, and then subsequently super infected with the HIV. So first we infect with the TIP, and nothing happens. And then we super infect with HIV, and I'm stopping the movie at 15 hours, and you can see HIV is starting to come up. All the cells have TIP, so they all should be expressing blue, but they're not expressing yet. Only after this 15 hours, now I'm going to let the movie go, the tip gets activated because it requires HIV and takes over. And you can take the supernatant off of this movie and throw it onto a new culture. We did this in the bioreactor setting. The percent of HIV infected cells is shown on the y-axis. So HIV on its own, without the presence of the tip, just in a regular culture, will take off and destroy all the cells in about 3 weeks in this reactor setting. And if we introduce the tip or take that supernatant off, we can keep the culture essentially healthy. Low levels of infection. It's not cleared, of course. This is not a cure. This is really a biocontrol. It can just keep the levels very low. We think that this blip over here is some of the first evidence we saw of coevolution of the TIP and HIV. The HIV tried to escape and then was knocked down again. That's usually a signature of coevolutionary dynamics. I'll show you some more in the monkeys. So this was some of the first evidence that we could build an interfering particle that knocked the virus down indefinitely. And so we went to first to mice, but then to monkeys. So the leading model for testing new interventions in HIV is really the rhesus macaque. SIV is a cousin of HIV. It's highly pathogenic in these animals. A model we used, in this model that we used, the virus kills the monkeys in about 17 weeks. So, and a very hot, very pathogenic virus. So we infect the monkeys intravenously, and the viral load, SIV viral load, is shown on the y-axis, time in weeks on the x-axis. The animals go to very high viral load of about 10 to the 7, 10 to the 8 copies per mil of blood, and succumb to their infection in about 17 weeks. So we gave these animals a single IV infusion, or IV injection actually, of a TIP lentiviral vector, either before or after infection. We did it both ways. And this is what we saw in the viral load. So these animals had about a 4 log decrease in viral load. It was a single injection, and the viral reduction was sustained. These animals lived for greater than 6 months, and then we sacrificed them for toxicology and other experiments. And we can analyze the tip copies because there's a barcode in this tip, and through a ddPCR approach. And even though the tip was only given once, either prior to or just after infection, a week after infection, we could detect the tip out to 6 months, which is really the evidence that it's being sustained and conditionally replicating together with the virus. So 4-log decrease, as I said, viral load is the major and primary determinant of time to progression to disease. So a 4-log reduction in viral load is a very effective reduction. If we could do this in patients, in human patients, that would be very effective. Human patients don't have viral loads of 10 to the 8th. Their viral loads are more like 10 to the 5th. So we only really need to get down below 10 to the 3rd. That's the clinically relevant threshold for transmission. Can we do that? We think we can, but we're still testing that. Part of the reason we think we can do that is we've looked in patient cells. These are cells from untreated patients. This is from Stephanie Strathy at UCSD. These are patients from a injection drug use cohort. And we can knock down virus in these patients, in patient cells effectively with TIPS as well. So we took this data to the FDA, and they were quite positive. We have TIPS in GMP manufacturing that's complete now, and a funded phase 1 clinical trial that is beginning imminently. Okay, so this is just some of the modeling that we did to show that TIPS can reduce viral load below 10 to the 3rd, which is the clinically relevant transmission threshold in patients. So now I'll go and I'll switch gears a little bit and show you that this concept works for other viruses as well, not just HIV. I didn't show you the Zika virus work. It's published though. So in March 2020, the DoD, which is funding our HIV TIP clinical trial, came to us and asked if we could repurpose the TIP technology for COVID, for SARS CoV-2. And I said, it took us 20 years to get it to work for HIV. There's no way we can repurpose it in time. But Sonali said, let's do it. And went into the BSL-3. Here's Sonali in her BSL-3 suit. And she's, as I said, really the hero of the story. So here's what Sonali did. Here's the SARS CoV-2 genome. It is much larger than HIV, about 29 kb, flanked by UTRs, 5' and 3' UTRs, and the genes, of course, are shown in different colors here. Sonali built a TIP, which is about 2 kb. It's an RNA, 5' capped poly-A-tailed RNA with the 5' UTR and the 3' UTR. And she used an M-cherry as a reporter inside of this TIP so we could see it. She also built a whole zoo of control RNAs for the reviewers, which were size matched, about 2 kb, also 5' capped and poly-A-tailed, but uses a luciferase instead of the 5' and 3' UTRs. So these don't have the UTRs. And we packaged these RNAs into lipid nanoparticles, essentially equivalent to the COVID vaccines, the Pfizer and Moderna COVID vaccines. These are solid lipid nanoparticles, 4 lipid mixes. And these then were formulated into an intranasal formulation and given to Syrian golden hamsters, which are the leading model of pathogenesis of SARS CoV-2. So this is an intranasal administration. It's a single dose. And what the experiment looks like is it's a 5-day experiment. The animals, it's very highly accelerated in the hamsters. So they start recovering after 5 days. We can give the animals on day 0 a very high dose, 10 to the 6 PFU of either Washington strain or any of the variants of concern. Delta is one of our favorites. It's highly pathogenic. And then the TIP is given up to 12 hours after infection. That's usually what is used also for other antivirals like molnupiravir. After 12 hours, you don't see much of an effect with those antivirals either. So 12 hours is the number, in part because the animals start to develop their own antibody responses at about 16 to 20 hours. So 5 days later, in the animal's lungs, in the hamster lungs, this is what the viral load of SARS CoV-2 looked like. There was about a 4-log reduction, a 3-log reduction, so highly effective TIP-reduced viral load. There was reduced inflammation, which we know is a major contributor to pathogenesis of the disease. Here are 2 pro-inflammatory cytokines. And if we did histopathology on these hamsters, so on the left here, I had to learn to read these histopath images, but the alveolar sacs are shown here. These astrocies are essentially pulmonary edema. So typically alveoli are supposed to be clear, oxygen exchanging cavities. And in these animals, they're filled with fluid or blood. These animals are very sick, probably near death, but the TIP-treated animals had very clear and open alveoli. So these lakes of blood, I call them, are indicative of severe pathogenesis. So this is just a 1-slide summary of what we found, and then I'll go a little more into detail. We saw this effect, but the question was, how is this working? Is it what's the mechanism of action? Many people had thought that the way that these defective interfering particles would act would be through inducing innate immunity or adaptive immunity even. So one of the things I'll show you is what we tested is that whether the TIPs induce or activate innate immunity. We're delivering RNA. Shouldn't the innate immune sensors be activated? We don't see that. None of the cytokines, pro-inflammatory cytokines we looked at were statistically increased between the control RNA and the TIP. It looked like interferon 1, IFN1, was maybe increased, but it was not significant. So what we looked at were interferon stimulated genes downstream and the upstream RNA sensing genes or signaling genes. So the interferon stimulated genes, INGs, were not increased by the TIP RNAs compared to control. And the TLRs, which are the sensors or RIG-I, were also not increased or activated. The reason for this is actually well known. If you go back even to the original Drew Weissman-Carrico paper from 2005 on the report of pseudouridinylate, you'll see that they mention that if you 5' cap and poly-A tail the RNA, you get a very similar effect to uridinylate. So it's known that 5' capped poly-A tailed RNAs don't induce an innate immunity response. It's RNAs that are uncapped or untailed. So this made sense that we were not inducing innate immunity. So if that is not how the TIP is working, how is it working? Well, the hypothesis was, of course, this R naught greater than 1. It's competing with viral proteins and packaging itself into VLPs and propagating with R naught greater than 1. So to test that, Sonali did this classical molecular competition assay. This is her specialty. She's a molecular virologist par excellence. So the RdRP complex is the viral polymerase of SARS CoV-2, and you can see that if you add TIP RNA, you can compete away by shift assays. You can compete away the RdRP complex at higher TIP, but the control RNA cannot, and the nucleocapsid protein also can be competed away. So what we believe is that the TIPs are competing for nucleocapsid. They're binding the capsid and taking it away, stealing it from the wild type, and also the replication machinery. And that's how they're lowering viral load. To test whether you can actually build particles and transmit with an R naught greater than 1, Sonali transfected the RNA, the TIP RNA, into cell culture once, and then continually passage in the presence of SARS-CoV-2 for 3 weeks. And this is what the data looks like. Even though the TIP was only transduced once, you can see a sustained 2-log or more to 3-log reduction in viral RNA or PFU. Sorry, this is actually infectious particles or plaque forming units. The control culture actually increases in infectivity. We see many people see this in tissue culture for SARS-CoV-2. It's loss of the furin cleavage site. So the virus adapts to cell culture, loses the furin cleavage site, and gets better at replicating in cell culture, but terrible outside of cell culture. The TIP-treated culture stays reduced. If you take the supernatant off of this culture and measure it for TIP RNA or viral genome, you see there's you can detect TIP RNA even 3 weeks after the original addition. The only way for this to be there is you can calculate the dilution factor. The only way is for the TIP to have been replicated continually by the wild type. We measured the efficacy by the IC50, for example, in these for the different isolates for the archival or original Washington strain, and then Delta, Omicron, all the others, and it's about 100 micromolar is the IC50, which is very comparable to the other antiviral drugs that are on the market. So within range. It also appears that this TIP is robust even out to the 2003 strain of SARS-CoV-2. This is some preliminary data that the TIP can compete for the SARS-CoV-1 2003 strain N protein. So we think there's a very high barrier to the evolution of resistance. And then just recently, it's a 2022 paper, not 21 paper. We asked whether TIPs could reduce transmission. As you likely know, the data on whether vaccines reduce transmission is mixed. I think there's some evidence that transmission is reduced, and quite a bit of evidence that it maybe isn't reduced. In other viruses, such as influenza, we have good evidence, unfortunately, that transmission is not reduced. And for other mucosally transmitted viruses, vaccines do not seem to reduce transmission. Antivirals, such as Tamiflu, for example, also do not appear to efficiently reduce transmission if taken late. So we asked, because the TIPs are acting locally in the nasal passages, could they be reducing transmission? Because the infected animal is, of course, spewing out infectious particles of SARS-CoV-2. And in the presence of TIP, there's a reduction in that viral load. So perhaps TIPs would be effective to reduce transmission. And so we did these co-caging studies where we put a naive hamster in the same cage as an infected hamster and measured first the shedding, the nasal shedding from the source animal in gold here. Over time. So you can see the TIP-treated animals in magenta here have a reduction in viral load, very significant, and it essentially is undetectable by day 5, whereas the untreated animals are still shedding large amounts of virus at day 5. So it seems to be reducing shedding. And if you look at the contact animals, they have a substantial reduction, a very significant reduction in virus titer. They still got infected. We think that's because the way we did this first experiment was without a barrier. So they were crawling all over each other. These hamsters lick each other and groom each other. So there's extensive fomite transmission. We did not see any transmission of the TIP. I could tell you about why if you're interested, but we still did see transmission in these animals of the wild type virus, but it was much lower, and these animals were much healthier. So I'll basically end there and just summarize what I've told you about. I told you about this concept of therapeutic interfering particles, or TIPs. These are a subset of defective interfering particles with an R0 greater than 1, which makes them a competitive molecular parasite, which we believe can enter a co-evolutionary arms race with the wild type. I showed you proof of concept for HIV TIPs in rhesus macaques. And then in SARS-CoV-2, the TIP, which is single administration, intranasal, and Robusta variants. And for SARS-CoV-2, there's also a funded phase 1 trial, and the IND studies for that are underway. and just a picture of our GMP manufacturing facility that we built. And I will end there and take questions.
Great. Thanks a lot, Leor.
Thank you.
Really fascinating talk. Really appreciate it. Let me say to our audience that there are two ways that you can ask questions, and of course, I encourage you to ask questions. One is you can chat those questions in using the chat function, and we'll get to them as you chat them in. The other one is if you would like to raise your hand, we will recognize you and unmute you, and you can ask your question orally. So let me strongly encourage everyone to ask questions and to get Leor to reveal some of the things perhaps that were unclear to you, or a little bit more about some of the things that he alluded to but didn't have time to talk about in his talk. I wanted to just, when you started the talk, you said, well, if you just eliminate all the open reading frames, and you end up with this little replicating RNA, that was the concept. But it didn't turn out that they didn't have an R naught greater than 1, and so I guess I know you found some that did, but why do those have R naughts greater than 1? Why do those particular ones have R naughts greater than 1?
Yeah, that's a great question. We're still trying to track that down, actively studying that. We have some ideas on the mechanism of how the R naught greater than 1 is achieved. So for HIV, the story is very different than for SARS CoV-2. For SARS CoV-2 and the Sabricoviruses, it seems like the UTRs are pretty much sufficient. For HIV, there appear to be these cryptic packaging-like signals or other sites that are throughout the genome that we were missing when we just took the LTRs. We found one of them in the CPPT, but even that we had to do some other things in order to get it to finally work. I think we picked for lentiviruses for HIV, we picked the hardest target first. We picked a target where there wasn't even a dip that was a bona fide dip that was described in the literature. And we think we know why. It's because the virus really has worked very hard to encode its cis elements, which are required for being transactivation or for being transcomplement. Its cis elements are embedded within and flanked by trans elements like genes. So you have to cut them out and do some genetic acrobatics to paste them next to other cis elements to get it to work. So that's a long way of saying, for some viruses, just cutting out the trans elements seems to be enough to get you an R naught greater than 1, and for HIV, the cis elements are in other places, and we have to find them and paste them in very specifically.
Mm-hmm. Okay, great, thanks. So we've had some questions come in, Leor, while you were chatting, and so Jim Thomas has a question. Might we expect the TIP to transmit in other examples, i.e., is SARS CoV-2 a specific case that doesn't transmit, or will that always be the case? I guess that's referring, if I get that, it's referring to your non-transmission finding with the hamsters.
Yeah. Great question. So the difference between HIV, which we think should be able to transmit, and we have some evidence that the TIP does transmit, at least blood transmission, and SARS CoV-2 and other viruses, is really their mechanism of replication and their life cycle. So the fundamental difference is retroviruses integrate. When they integrate, they will become lifelong in that cell. And that cell then has a window of its entire lifetime to be super infected. So for HIV, you can infect, in principle, very few cells with TIP or transduce very few cells with TIP. And if any one of those cells in that small population gets super infected by HIV, at any time that that cell is still alive, you will restart the TIP transmission or mobilization from cell to cell. In the case of a virus like an RNA virus like SARS CoV-2, the TIP transduces that cell and the RNA has a lifetime of 2 hours, 6 hours. If it doesn't get super infected by the wild type virus within that short time frame, that cell is no longer a DIP transduced or TIP transduced cell, and it becomes a regular susceptible cell. So that's the major difference, is that we think we don't see transmission of the TIPs in the RNA viruses because there's such a short amount of time for any individual cell to be super infected. And if the TIP gets in first and it's not followed directly by a SARS CoV-2 super infection, then it's like the TIP's not there anymore. So you have to have incredibly high viral loads of TIP to get to many cells, followed almost concurrently by wild type virus infection. Whereas in the case of HIV, all you need is the TIP to get in there, and then it integrates, and it's in the genome, and it's there forever. And any time after that, HIV can super infect. And that's why we think we see in one case transmission, and the other case we don't see transmission. It's these bottlenecking effects. I don't know if that was clear. Does that make sense?
Yeah. Well, I mean, I think I got it, and if I got it, I have a feeling that Jim Thomas got it too. So Mike Santos has a question. He says, "Thanks for the presentation. Have you modeled how TIPS would work in the context of sustained ART followed by treatment interruption?"
That's a great question, and that's in fact the basis for the clinical trial. All clinical trials have to be these so-called, these days, analytical treatment interruptions, or ATIs. And that's exactly how we're running our clinical trial, as an ATI. We're doing it in this is a first in human, of course, an FIH trial, and it's being done in an end-of-life cohort in San Diego that Davey Smith runs, called the Last Gift cohort. These are patients with a terminal cancer diagnosis who are HIV infected, and many of them, over 50% of them, discontinue drugs even in the absence of a trial, because it's not the HIV that kills them, it's their metastatic disease, their cancer, that kills them. So many of them come off their drugs anyway. And that is how we're going to be testing the TIPS in this cohort of patients. And then subsequent trials, phase 1, 2 trials, will also be analytical treatment interruption trials, where patients discontinue their antiretroviral drugs and are given TIP. We've done a little bit of this in mice. They're very challenging models to do it in, and it's possible we will do this in the monkeys as well. Yeah. So I'll give you a little more context. Antiretroviral drugs halt infection, so halt HIV infection very effectively. So the TIP will not be able to really do anything because it has no raw materials in order to parasitize. So it has no raw materials in order to replicate. So we have to discontinue therapy in this case. And in other drugs, you have to discontinue therapy, drug trials, because otherwise you won't be able to test the efficacy of the drug. There are some specific issues that we need to keep in mind, which is some of these antiretroviral drugs, for instance, reverse transcriptase inhibitors, have long half-lives. And if we have residual reverse transcriptase inhibitors in the system, the TIP won't even integrate because of the residual reverse transcriptase inhibitors. So we have to discontinue, and then a time later introduce TIPS after discontinuation of therapy, so that we're sure that the drugs have been cleared. There's a similar problem for other gene therapies for HIV, that people have a very similar problem with reverse transcriptase inhibitors.
Okay, great, thanks. Let's see. I'm gonna get to Andrew Kagundu here, who asked a question, and I'm gonna let you take a shot at this. So he said, I joined late, and I'm still missing why tips for treating HIV and COVID are considered to be gene drives. So, I'm laughing because I know we had a conversation the other day about this. So why don't you go ahead?
Right. I mean, I don't really think they're gene drives. I think there are some similarities to gene drives. The tips are conditionally replicating; they mobilize, and I think maybe the most similar or in some way analogous to the gene drive idea is that they have the ability to, at least in principle, co-evolve and establish a co-evolutionary arms race because of their replication potential, conditional replication potential. So they continually mobilize with the wild-type virus, and they're continually co-evolving to maintain their selection advantage or selection pressure on the virus. They're under the same selection pressures, and they're potentially transmitted with the same through the same routes of transmission.
Yeah, I mean, clearly these are we're not talking about the kinds of genetic drive elements, selfish genetic elements that intragenomic selfish genetic elements that we're talking about here, but. So I'm speaking to Andrew now as we talk here, but we are talking about self-replicating entities that can spread through populations that then have an effect on. In this case, they have a modifying effect on the population which receives them. So in a very broadest sense, they have that. And but I also want to point out that Leor, it was your NIH proposal that used that word that got me hooked up with you to begin with. So you are responsible for
one way that I have presented it. When people ask what's the similarity between gene drive and TIPS is I think of gene drives, the canonical gene drives as vertical, transmitting, through the population vertically. And I think of TIPS at least in the HIV sense, where they do we think they can transmit between individuals as horizontal. So a form of a horizontal gene drive in contrast to a vertical canonical gene drive.
I mean, I see that for sure. Although selfish genetic elements move horizontally as well, but not on the same scale. But so I take your point for sure. Ethan Beer has a question about could TIPS be used for influenza?
Good question. There are groups that are actively trying to repurpose or to develop TIPS for influenza. When we started the DARPA program many years ago, so about 2014, 2015, many of the groups that they funded, the Intercept program, were influenza groups. And at first, I was a little bit surprised that we had worked with them for many years, and we thought they were going to fund our HIV work, and they did. And they also funded many other groups, about 20 groups around the world, and I think 15 of them were working on influenza. But I think it was good. I think it was an important service to build this field, and they funded so many of these academic groups to try this in many different viruses, not just influenza, chick. The big ones were probably flu, chikungunya, and then people moved over into SARS and MERS and other of the sabrocoviruses. So in principle, it could be used, I think, for any of the viruses. There is actually there was in the early 2000s, mid 2000s, not early 2000s, a group in the UK who was developing a defective interfering particle for influenza. The problem was that it it worked through innate immunity. And so it was really not a defective interfering particle. It was a defective virus that activated the innate immune system. There were a lot of questions about safety with that approach. And so the company, as far as I know, went under.
Okay, let me continue to encourage people to ask questions or raise your hand. Be happy to recognize your hand as well. I do want to touch on, get your thoughts on some of the sort of social implications or some of the governance types of issues that arise with this. Certainly within the gene drive space that we're talking about, that we talk about all the time with, say, modifying mosquitoes and releasing those into an environment and having that drive spread through populations. It's an idea that generates lots of discussion around governance and ethics and things like that. And this would seem, I'm sure this is coming up in your space as well. So could you just say something about that, and what some of your thoughts are, what some of the reactions are within the community about these sorts of things?
Absolutely. I'm glad. This is part of the reason I really wanted to talk to this group. I wanted your input. We have, over the years, engaged with bioethicists, in particular at the University of Chicago and at other institutions, but also part of the reason that I agreed to give that TED talk was to socialize the idea. I think it early on when we first proposed this in the early 2000s, it was highly charged. I think the idea of anything transmitting through the population was just anathema to many people. But the bioethicists did not seem to have an issue with it. In fact, they pointed out that many live attenuated vaccines, this was part of the discussion, and in particular, there were bioethicists who were involved in oral polio vaccine. WHO, the WHO's decision to adopt the live attenuated Sabin vaccine, which was known to transmit between individuals, and they made a very strong case that this was a recognized benefit and part of the argument for choosing the Sabin vaccine over the Salk vaccine. There is literature on this, so I think they have a good. I believe them. I think there are perceived ethical issues for this type of approach. Consent issues. There are the same consent issues, or I think actually much worse consent issues, I think, for live attenuated vaccines, which transmit indiscriminately. In the case of the HIV TIP, if it does transmit, it will be through, fluid exchange and very circumscribed transmission in the high-risk populations. So with the discussions, and we've even had a conference on ethical implications of TIPs and related technologies. The ethical considerations appear to be circumscribed compared to live attenuated vaccines, and the consent issues have already been dealt with. You have to advertise to the population that you're treating. And you have to educate the population that you're treating and provide alternate means of transmission control, PrEP, for example, in the case of HIV, for partners of treated individuals. But that's my perspective. I would love to hear other perspectives.
Yeah, well, I mean, certainly within the insect space, the idea of consent and to who grants it and so on has certainly come up, and governance of these technologies is still an ongoing discussion within the gene drive space for, say, for malaria interventions in Africa. Yeah, it just seems to me that the issues with an HIV TIP might be a little bit different than a mosquito that's passing on an anti-malarial gene or something. Jim Thomas has another question, and that is, says you are DARPA funded, and this has obvious dual use potential. E.g., rather than express GFP, you could express something else. How is that being discussed? And also, is this approach being developed for plant viruses and agricultural use?
Okay, so the dual use idea, that came up early on, I would say in the early 2000s. And it occasionally does come up. I would point out that you could encode something else in the TIP, but even when there's two answers here. Even when we encode GFP, and this is a problem in the entire synthetic biology space, even in cell culture, that anything that you encode that doesn't provide a selection advantage for the synthetic entity, even if it's neutral, it's lost. So even GFP, which has a very, very slight, maybe cost, metabolic cost to the bacteria, even the synthetic biologists have found that gets spit out within 3 or 4 generations. So unless there's a selection advantage, things get lost, and that's a major challenge that we have in the synthetic biology space. And bacteriologists, the synthetic biologists have same thing. When we put GFP into the TIP and put it in a monkey, the GFP is useless after 3 days. We don't see it any longer. It just gets immediately silenced, or probably stop codon mutations is our guess. So if you put something with a negative selection, right, with a disadvantage, something nefarious, it's almost certain that's also going to get lost. You could think about putting some sort of entity which has a positive selection pressure and would also be dangerous. And again, I think the problem becomes that this is dependent on the wild type virus. So you're not introducing a new entity which spreads on its own. It's really only spreading within the population of infected individuals. So dual use here to me seems, I guess it could be dual use, but it'd be a very, very limited dual use program targeted against individuals who are infected, and only when they're infected. Yes, but really quite not a huge bang for your buck dual use, I think.
How about the agriculture part? Plant viruses.
I think it's a really good idea. We personally, my group, has not focused on plant viruses. Sonali, who did much of the SARS CoV-2 work, all of it really, came from a plant virus world where defective interfering particles are well known and studied. I don't know of much research around their use therapeutically or as biocontrol agents there, but they're very well known in that space.
Great. Okay. I think we can just about start wrapping this up. Before we do, I wanted to let those who are still in the audience know that this is the third in a series of 6 webinars, and our next webinar will be on April 19th in this series, and it'll be by Mauro Rebello from Brazil, and he's going to talk about his early stage development of a gene drive system for an invasive mollusk that is a severe pest all over the world, the invasive golden mussel. So I'll just say that. I think, look, Leor, I just wanted to say thanks again for participating in the webinar series. It was really stimulating and really enjoyed it. And I'm sure everybody that was in the audience really enjoyed it as well. So
thank you very much.
Yeah, thanks again. I hope we get a chance to interact sometime in the future as well. So with that, I'll say thank you for the attendance at this webinar and hope to see you at our next one. So with that, I'll say have a nice day.